this year, one of the things that I wanted to hear more about at this session was food safety. A lot of y'all are probably aware, we've seen some food safety issues in onions in recent years, and we just want to maybe try to stay ahead of the curve a little bit in Vidalia, make sure we're keeping our onions as safe as they can be and growing them the way they need to be grown and handling them the way they need to be handled. So I've got a couple of food safety talks this morning for you. And our first one is Dr. Laurel Dunn, professor with Department of Food Safety at UGA. She's going to talk about some work that she did. Actually, she did some research at the Vidalia Onion Research Center looking at E. coli survivability and different like chicken litter and compost and things like that. So I'm going to let her tell you the results of that study, and then we've got a talk about some stuff that involves the inside of the packing shed. So, Laurel, if you want to come on up.
All right, thank you. So, one thing I want to just make clear starting off, this is not one of those food safety talks where I'm going to be fussing at everyone. So I just want to provide you with some research updates that have been going on. And actually, this was, a lot of this came from a talk too that I actually gave yesterday, but a colleague of mine had been supposed to present, Keith Schneider at University of Florida. So you're going to see the IFAS logo as well on this, because this is work he and I have been doing together really since I was a postdoc at Florida, and then we've continued working together once I moved to UGA. And now one of his former students is in my lab, and so it's just, I realized his presentation was really a lot of projects that he and I have done together, and we're going to finish up with some of the research that we've been doing in Vidalia. So why we're concerned about this is, as you're aware, I know, we've been having some outbreaks in onions, and it's kind of a new commodity to be having some of these issues with. A bit of a surprise in fall 2020. We had some other stuff going on at that time that we were a little bit more focused on then, but we also had our first kind of outbreak with this flavor of salmonella, if you will, that actually pretty much sickened an American in just about every state, and a lot of Canadians as well. And this came from some domestic product. They never really found the smoking gun for where it came from, though. So there have been theories that it contaminated irrigation water, but really haven't totally traced that back well. Pretty much in fall 2021, we had some more issues, and then we had this interesting outbreak that we're just kind of wrapping up with diced onions. And what's interesting here is, so we say we don't have problems with onions really because of a lot of the compounds in onions, those isothiocyanates that are naturally antimicrobial. You'd think with a diced product, that would be a safer product than a full bulb where you've not exposed those, and yet we've still had some outbreaks with this product. Again, not really entirely sure what happened there, but they did find salmonella in the environmental farm samples that were collected. And we had multiple strains of salmonella involved in that, which usually is an indication that something went bad wrong if we're having multiple types of it. And I would think maybe an animal source, a soil amendment source or something like that would be causing this. So there are a few things that could be going on here, and we're just not sure. So for one thing, we could be having some changes in the pathogens themselves that are making them more robust. So I remember, when I was making cookies with my mom in the kitchen 20 years ago, it was always, don't eat the cookie dough. Why did I not want to eat the cookie dough? It was salmonella in what? In eggs, all right? Now, when we're making cookies, what's the reason you're told not to eat the raw cookie dough? The flour, because we're now finding E. coli in flour, and it's making people sick. So our understanding of pathogens changes, but sometimes the pathogens themselves change, and some of the resistances. Our improvements in the ability to detect pathogens are improving, and our ability to trace things back. So, maybe this is a thing, we've been having issues with onions, we just never knew because we weren't able to detect where they were coming from or to trace things back. And then maybe we just weren't looking for issues. We weren't really worried about produce 20 or 30 30 years ago because we always demonized ground beef if we had an E. coli outbreak, or we demonized chicken if we had a salmonella outbreak. We really just weren't looking at produce because we didn't think there were issues. So we're just going to jump into this, and we've already kind of covered our onion outbreaks, but we're going to go into some biological soil amendments of animal origin and what that means for the FDA. We're going to discuss some pathogen prevalence and studies that we've conducted at UGA and at UF, some pathogen survival in soils, some of our effects of our soil amendments, and then just some best practices. I can't have a food safety talk without giving you some recommendations on what I would do if I were more involved in growing on a farm. So when we say biological soil amendment of animal origin, has anyone in here been in a Produce Safety Alliance grower training? All right, we've got a few hands. So you're familiar with this term. This is pretty much any animal-related product that you might use to amend soil. So manure is the one we tend to think of the most often. It could be animal mortalities, fish emulsion, things like that. It can be table waste, used alone, or if we mix it in with other compounds. We call this a biological soil amendment of animal origin. FDA is most concerned with these if we're not treating them in some manner. So we can use raw poultry litter. We can use raw manure straight out of the cow, but FDA really likes to have some sort of control, like composting or some sort of treatment in order to reduce our prevalence of pathogens. That's not always going to happen, right? I mean, if you're scraping out a chicken house and you're trying to get rid of that material quickly, it's easiest just to move it once, spread it, and be done with it. So because of this, USDA has said, yes, you can use things like untreated chicken litter out of a chicken house, but they really want you to follow this National Organic Program guideline for the use of untreated soil amendments, so our untreated chicken litter, our untreated bovine manure, things like that. And so the recommendation is if you're going to apply it for a crop that is going to grow up and not contact the produce, they want you to have 90 days between when you apply it and when you're going to harvest that. So if you're applying it to citrus or something like that, you need to wait 90 days before you harvest it. Something like onions, because it's contacting the soil directly and it's going to be contacting that soil amendment, they recommend that you wait 120 days between when you apply that and when you actually harvest your crop. Now, we're going to discuss some studies that we've conducted, kind of looking at that and seeing if that's a reasonable recommendation. So first, this was how I kicked off my postdoc with my newly minted PhD down in Florida. They said, great, you're here, you're going to start collecting poop samples for us all over the Southeast. So we did this for both cattle operations and for poultry operations. And we were looking in this case for E. coli in bovine manure. We visited 25 farms, and out of those 23, we had a 92% positivity rate. We were collecting samples out of the manure piles and finding E. coli in those. Probably not a shocker. I don't know that anyone's really shocked that we went and sampled cow poo and found E. coli pretty frequently. But, you know, we needed some evidence to support that, yes, if we're going and finding it, if we're going and looking, this is where we're going to find it. We also conducted a couple studies just doing the same thing, again, with poultry litter. When I was a poor little postdoc, we got this work done where we found about 20% of our samples were positive. When we were looking at chicken litter, we're positive for salmonella. Keith's lab conducted some work after I left and found that they had about 55, trying to do math on the fly, 60% positivity rate when they were sampling farms. A lot of this just comes from where you're collecting this. I was collecting a lot out of dry stack sheds where things have been sitting for a while and kind of naturally getting hot and going under a natural composting process, even though FDA wouldn't call that composting, but it was still happening naturally. I think they were checking out more fresh samples straight out of the chicken house for this. So we've established these pathogens are in animal feces. Don't think anyone's really shocked with that. But then what Keith's group did is they inoculated these microcosms with E. coli, or they were following, they got manure that naturally had E. coli in it, and they put it in these soil microcosms at different temperatures in the lab. So they got soil from 2 different locations, and they stored it at 2 different temperatures. And what they found was that at a higher temperature, the E. coli tended to drop off pretty quickly in these soil microcosms that just had natural E. coli that had been added into them from the manure. So after storing them, they start at a pretty high level, and by about day 200 or so, they had died off in these microcosms. At 20 degrees, though, that's these upper lines here, they got recoverable E. coli out to almost 400 days. Now, what were the NOP guidelines for onions? It was more 120, right? So this is indicating that we can still get pretty extensive survival of some of these microorganisms in our soils. And as you can see here, this is where it wasn't until these points where we started to see some really rapid die-off. So then they wanted to see if moisture level impacted the survival. And so what they found here is usually if things were a little drier, this 15% moisture, that's that top line, a lot of our microorganisms could persist in the soil a bit longer. And again, they're getting recoverable microorganisms out to day 200. So they're still able to recover things well beyond that National Organic Program guideline. So then moving forward, they took these plots, this is in live oak, where they amended them with bovine manure, and they were just tracking the naturally occurring generic E. coli and monitoring over the course of 300 days. What they found was in core samples, that's what this chart is, and these are our surface samples. What's interesting here is in the fall months, they got a really rapid decline. So by about day 25, 30, a really significant decline of this E. coli in these soils. But as you can see, out to almost day 300, particularly with these heavy inoculated, that's what the gray is, and these like medium inoculated plots, they would still get E. coli popping up. Now, this could be due to a lot of factors. If they'd gotten a rainfall, if there'd maybe been an application of something to the field that these microorganisms were able to use to grow. So I think the moral of the story here is we still get a lot of persistence with these really low levels that could pop up. And I think that's how some of our groups have had kind of some bad luck with using these amendments where they've applied them, they've probably been following that guideline, and then they've had some persisters kind of pop up. So then this was also conducted in the summer, and I just want to point this out. That last study was done in the fall. They repeated it in the summer, and they found that they got a much faster die-off and didn't get the same level of persisters popping up afterwards. So seasonality probably plays a role in this, where when you're getting really warmer months, you might be getting faster die-off of some of these microorganisms, and these particularly microorganisms of public health concern in our soil. Keith's theory on this, I really like this theory, so we'll go ahead and discuss it, was just that some of this extended survival, or I guess this rapid death as the temperature goes up, could be maybe due not to the fact of the heat itself in the soil, but could be due to the fact of when you have warmer temperatures, you have a more robust soil microbiome, you have a lot more microbial activity, a lot more metabolic activity going on in that soil, and maybe those E. coli are being outcompeted, consumed, starved out of the system. And so what they did is they conducted this microbiome study where they sterilized soil. And then they inoculated it with 3 different flavors, if you will, of pathogenic E. coli. So we have our sterilized soil up here, pretty much, and then and sterilized, I use loosely, but they had killed off a lot of the microbial community, and then they just had straight soil here that they inoculated the E. coli in, and decided to see how long these microorganisms could persist. What they found was the different strains of E. coli didn't matter. They were all statistically similar, but in the samples that were autoclaved, there was less competition for that E. coli, and they got extended growth out there. Extended growth beyond that. Here, we have it, yeah. So we have, particularly at day 30, significantly greater growth in that autoclave soil. So kind of supporting this theory that maybe it's not just straight temperature, but maybe there's something else going on with the microbial community that's suppressing some of that E. coli growth. But one thing I want to point out, as microbiologists, we get really excited when we find one organism, and we will start to assume that it behaves similarly to a lot of others, and we'll use it in studies. We call this a surrogate because we think it behaves similarly. So we've been doing that with E. coli and Salmonella quite a bit, and I am involved in a lot of studies where we use E. coli, but kind of are trying to use it in a spot where we think it's similar to Salmonella. They conducted the exact same study with Salmonella, and they found that it did not matter if they removed those natural microbial colonies. If they used sterile soil, the Salmonella died off just as quickly as if they used soil that had not been sterilized. So just a warning out there of, if you see some data that's published for one pathogen, if it is not the pathogen you're worried about, take that data with a little bit of a grain of salt, because we're here, we're interested, if we're talking about poultry, if we're applying poultry litter to our fields, we're probably most interested in Salmonella. A study describing E. coli survival might not tell us exactly what we need to know about Salmonella survival. So now this is getting into the study that we conducted in Vidalia. This was a study that we conducted concurrently. So I was working with Chris, and Chris grew all of our transplants for us, both at our Georgia and our Florida locations for this. So we created these little plots here. If this does not look like Vidalia, there's a reason for that. This is actually our research station in Live Oak, the UF station that we were conducting this at, that Keith's group was leading. And so these plots we amended with composted poultry litter, with poultry pellets. We didn't add anything to, or we may have just added a synthetic fertilizer, and then we inoculated them with E. coli and then planted onions in them. And what we were interested in seeing is how long is that E. coli going out? If we assume that we're applying our chicken litter, like right before we put our transplants in the ground, are we still, by the time we're wanting to harvest those onions, are we still going to be recovering any of those microorganisms out of the soil? So here was our data that we found out of Vidalia, and I have it for both locations, so we'll get to the Live Oak data too. But what we found was we got some tapering off here. This gray line indicates our soil that was amended with a composted poultry litter that then we re-inoculated. And so we found, we got a more, I guess, dramatic decline here. This was just our plain soil that would occasionally receive a synthetic application, but it received no actual organic material to it. But what was interesting about this is this red line is our heat-treated poultry pellets. And there is something really weird for anyone who ever uses heat-treated poultry pellets to be aware of, because we're starting to see this across studies. For some reason, these pellets really promote the growth of pathogens if they're present in the environment. We're not really sure what's going on here, and maybe not promote, but at least protect these microorganisms in the environment. So we can see, again, I want to point out this 120 days. This would be where we would say, okay, National Organic Program says we're good to go. We are still collecting, this would be about 10 colonies of this microorganism in a gram of soil in this period. Keith's group found the exact same thing. You always like it when you design two identical studies and plant your plants on the same time and are trying to make sure that everything agrees. So he found kind of the same thing with in this case, their composted material. By about day 65, they weren't finding any more recoverable pathogens. So occasionally we tend to see some of these persisters pop up. We still got some of those. But for all intents and purposes, we would have been fine following the National Organic Program in this case. But again, our poultry pellets have this weird thing where we're still recovering pretty high levels, in Keith's case, out to 140 days. So just following the NOP guideline might not be sufficient to reduce risk, particularly if you got some event here, like weather, a rainfall event, or something that might enhance the survival of any pathogens that might be present in that soil. And I always forget when animations are put on the slides, so that's why we're getting the late arrows. So one thing Keith wanted to look at, and so he had a graduate student who is now my graduate student work on this, and so they were interested in seeing if maybe synthetic fertilizers, play a role in some of this die-off, this reduction of some of these microorganisms in the soil, particularly because in most of our production instances, we might apply poultry litter before we transplant. We're not going to continuously be applying that poultry litter, though. For fertility throughout the season, we're going to be applying some of these synthetics. I think what's fascinating with this is we're just going to jump down to the end at day 56. These lines on the bottom, these kind of bluish green lines, are either all synthetic fertilizer, so no organic application, or poultry litter or poultry pellets where they added a synthetic fertilizer then throughout the season. And they got a much more rapid decline, or at least by day 56, they weren't recovering these microorganisms anymore with the addition of the synthetic fertilizer. So just an interesting thing, again, they have their poultry pellets up here. Those are our worst performing ones as far as promoting the growth of these microorganisms. And then up here is our composted poultry litter that was supplemented. So just some interesting takeaways with that. One thing, too, I guess with this study is we have not published this data yet, but part of our study was conducted, we were collecting these soil samples throughout the season. At the end of the season, we collected onion samples from these soils, and we did find a small amount, but still some transfer from the soil onto these onions that were being pulled out of these amended plots. So we were able to get some transfer in these. So now, because I can't leave you with some recommendations and give you the food safety spiel, FDA has I won't say no problem. FDA allows the use of untreated soil amendments like poultry litter. They allow that in crop production. They don't have any hard and fast guidelines yet for that now. They're not regulating that now, but they are recommending those National Organic Program guidelines. I think our research suggests that those guidelines are good to follow, but be aware, particularly with a root crop, where you're definitely going to be getting that direct contact with the soil, 120 days might not be a sufficient time for you to wait. That my recommendation would be, as soon after harvest as you can. If you're wanting to amend the soil, apply chicken litter at that point, and then just really maximize your amount of time and rely on synthetics throughout the rest of your growing period. But really try to extend that time out as much as possible in case you do have a load of litter that has an unusually high level of pathogens in it. If you can use compost, great option. I know it requires a lot more handling, a lot more expense, a lot more work, but we always like to see that. Another thing, I guess 2 more things I want to leave you with. We'll start with the first I don't have up here because I'll probably forget it. The FDA is really starting to do an increased focus on this concept of adjacent land use. So they're starting to see, they're not starting to see, but they're starting to connect, I guess, outbreaks that might be related to animal agriculture in the general vicinity. There have been some outbreaks where there has kind of been a smoking gun, like a CAFO or a really close poultry production, where they've been able to pretty definitively say this pathogen came from this animal production and was put in these produce fields through air transfer, water transfer, things like that. FDA is really starting to pay attention to that. I would just have you be aware of that and of what's going on your operation. It's hard to define, how close is too close. What is the FDA, where are they going to go with this? But I want you to be aware that that's something FDA is starting to pay attention to. Just be aware of what's going on in the environment around you. The FDA might be making some adjustments to the water regulation, that portion of the produce safety rule, where they're going to be very interested in you doing a full land survey, pretty much, or an assessment of the area around the water you're going to be applying for irrigation. So, thing I want you to be aware of too. And then this idea of clean breaks. And if you're not familiar with this term, this just means a full sanitation stop. So you process for a day, you close down shop at the end of the day, and you fully clean and sanitize your equipment. So that way you can prove, using a validated cleaning and sanitizing method that you have proven is effective on your equipment. This really helps limit the impact of an outbreak or a recall if you have one in your operation. Because if someone comes back and says, all right, we found that onions that you produced on this day may have been contaminated with something that came in from the field, or we got contamination in your packing house. You might say, yeah, all right, we did have a problem on that day. Something happened. But we can establish that we had these clean breaks where we cleaned and sanitized on either end of the recalled product. And you can prove by having this really good cleaning and sanitation that it was a limited problem. If you go your entire season without cleaning and sanitizing, and you get some onions or some product, it doesn't matter, not just onions, not just picking on onions, but you get some product that might be contaminated, you could have an entire season worth of product recalled. So I'm just going to leave this up for a second, or actually I've got the QR code on this next one. If you want to take a survey for us, we're trying to get some USDA funding about cleaning and sanitizing of harvest equipment. We'd love it if you would participate in that. And are there any questions? I guess I have a minute probably left for questions. Yes? Mm-hmm.
I saw that you brought it out all the way to 140, or you brought it out all the way to 140 days and still had colonies present.
Mm-hmm.
At what point did you stop trying to see that there was no colony present, and how far out did it go before you found the colony?
Yeah, so I think one thing that the graduate student was teased with who did this was you're going to keep doing this until you don't find any more in it. So the 140 for that was as far as we went, because at that point we had taken all the onions out of the field and we were done with that. Did I just answer your question?
I think you did.
Okay.
But I've got a quick follow-up on that.
Yeah.
You said there were some onions found.
Mm-hmm.
What was the percentage? Was it statistically relevant? I mean,
Mm-hmm.
poultry litter is used, or the pelletized is used a ton in organic in this area,
Mm-hmm.
whether it's Vidalia onions or something else. So I think that's a really relevant thing to look at across the board, where we're using it so much in this
Mm-hmm.
vicinity, to know that that's
Mm-hmm.
there and that it's going out that far and that we've got a higher growth rate and maybe that might make changes in industry use.
Two things I want to point out. One, and I should have made this more clear, us finding some of these organisms coming up does not mean that they're clinically significant levels. It just means that we're still finding them there.
They're living is the thing.
Yes.
That's the issue there.
And the other thing to keep in mind too with these pellets, that survival is going to be if you get a contaminated batch. There's no validated process for poultry pellets to that FDA level of, no one has yet validated that system. That would probably be a good project for one of my students to do. But until we do, we are assuming that if those come in contaminated, there could be an issue. If they don't come in contaminated, that could be a very safe route to go. But it's just something, it kind of goes back to if you have these, storing these in a manner where they won't be contaminated, making sure, pests are kept out of them, and being aware of what is being applied in the land around. And back to that adjacent land use, if you could get contamination in the soil and using pellets, there could be an exacerbated effect.
I'm just thinking to the point that if there was something previously on that land and if that product allowed for further growth,
Yep.
if that's going to be an issue, because it could come from flooding, right? We've had a lot of rain this year. It may not be directly adjacent land, but you've got ponds, there's E. coli and stuff, salmonella in the ponds, and frogs, et cetera, and else. So if that's allowing that pathogen to stay alive, if that extended, where a compost which isn't, is that something that we need to be looking at and paying closer attention to, just because of the ability for it to give a longer shelf life to that product?
Yeah, I think we're at the point we just need more information, but it's been pointed out now that we need to start looking at that.
How many onions did you say were found that are transferred?
I forget the exact number. It was not significant enough to put in here. We just saw that we did have some transfer of some. And again, it comes back to some transfer does not mean it would be enough to make someone sick or to cause an outbreak.
Real quick, with that
Oh, yeah.
transfer to those onions, was that before they were put into a drying room, or was that after they were put into a drying room?
Good question. That was before they were put in a drying room. So we pulled them, we left them out for 2 weeks, and then that was when we sampled those after they'd been sitting above the surface for about 2 weeks, but before they got dried. So yeah, that curing and that drying could further kind of help you out from a food safety standpoint of controlling those as well.
Carl, I have a question. You mentioned your theory about a more diverse microbiome might help, I guess, not necessarily reduce or stop.
Could outcompete some of these.
So a lot of Vidalia onions are rotated with summer crops.
Mm-hmm.
And we use and sometimes land is left fallow, but trying to do at least a rotational or cover crop between onion crops, would that lend itself to a potentially safer scenario based on what you're saying, or? I
don't know if it would help or hurt, quite honestly. I will say, and looking at some of the things I've seen Keith discuss on the topic, his whole thing is, if you increase some of this organic matter, you're naturally increasing some of these microbial communities, and so you might be getting just a more robust soil community. But what if you're also adding some of these pathogens to that soil community? So it's kind of, you could go either way with that. So I don't have a clear answer. I think it's one of those, it's complicated and it depends. But good question.
All right. Any more questions for Laurel? Okay, thank you. All right. Well, next up, we've got Dr. Neaton from UC Davis. We asked him to come and talk about some research that he has done looking at things we can do inside of a packing shed or packing facility that have a food safety relevance. So Dr. Neaton is he's in the Department of Food Science and Technology at UC Davis, and so he's done a few projects looking at food safety that we can relate onions to. So Dr. Neaton, I'll ask you to come on up and get started with your talk. Here's the remote.
Thank you, Chris, for the kind invitation. So I'll be talking to you, I'll be just following on what Laurel stopped at, where we're talking in the field. This is talking about what happens after you bring your product to your packing facility. I think I should move it to slideshow. That would be better. So we'll talk about cross-contamination risk in dry environments. And interestingly, there's a lot of data of cross-contamination in where water is involved. But when it comes to dry environment, there's not enough data out there that tells us that how does microbe move from product to surface or surface to product as it moves through the dry environment. And we'll also talk briefly about antimicrobial coatings that my group has been interested in for some time. Before I start, I'd like to acknowledge the people who did the real work. You should see she's a postdoc in our lab jointly with Professor Linda Harris and working on the cross-contamination project. The antimicrobial project is being led by a graduate student, Yun Bin Kim, and my collaborator, Professor Gang Sun, significantly contributed in it. The funding for this research was supported by Center for Produce Safety and USDA and AFA. So when it comes to post-harvest contamination and dry contamination or cross-contamination in dry environments, what we're looking at is we are looking at different commodities here. I'm going to talk about onions and I'm going to talk about peaches. I know onions and peaches are not the same. I'm not trying to tell that they are the same commodity, but they go through slightly different processes but have some similarities. Now, in case of onion, you almost go through a complete dry process as you get it from the farm. But in case of peaches, you actually have a washing step in between, and then waxing step, and then a dry handling step. So they go through a slightly different sequence of processing, but they do see a dry end of the process in this case. So funded by CPS, we have been looking into what happens if you have a contaminated produce, either a peach or an onion, that ends up in your facility. How would it transfer that contamination to your conveyor belt? Or how would it transfer that contamination from a product to another product? And then how would the microbes move from the conveyor belt back to your uncontaminated produce that may happen to pass after you have done the first inoculation through a contaminated produce in this case. And so we broke this question into two parts. The first question was, how long can a bacteria survive on a conveyor belt? Like, we need to know that answer because that is an important question. Like, if it can survive for a longer period, that's something to worry about, and we need to take care of it. If it survives and dies off very quickly, maybe that's not a big deal in some cases, and if we have adequate sanitation, we may be able to manage that risk. So that's the first question we asked. And the second question we asked is that what factors affect the bacterial transfer? You could have a pristine looking clean belt, or you could have belt with some soil on it. You could have belt with some soil and onion juice on it, or you could have some onion juice on it. Like, you can have different scenarios that you all see in your facilities when you are running the plant. So does that matter? Where the microbe is on the surface? So that's the second question that we are asking in this case. And the way we did this experiment, I just want to go into the details. We took two strains. We took a cocktail of Salmonella, so it's a 5-strain cocktail. This strain cocktail has been previously validated in various other dry environments, so it's a very good surrogate. I know Laura brought out a good point that, when we use a surrogate, we want to make sure that, we're using the right microbe system. So in this case, it's a Salmonella directly. We are also using Enterococcus faecium, E. faecium, which is resistant to desiccation, often used as a surrogate for thermal processing and in food safety analysis for Salmonella. So it's a good surrogate for Salmonella in this case. And food contact material, we're using polyurethane. The samples were obtained from industry partners, and stainless steel. And then we also did experiments where we did survival without onion extract and inoculated at 3 different levels. We call them high, moderate, and low levels. And then we also did survival with onion extract, where we used bacteria which was diluted in onion juice, inoculated on a surface at a moderate level in this case. And then we maintained the storage conditions, low humidity conditions of 34% and ambient temperature of 21 in this case. So first off, what we noticed here is the log reduction of Salmonella, starting with the high, medium, and low. What you notice is that on a plastic surface, which is a polyurethane belt, you find a difference. The microbes do get inactivated, but it depends on the inoculum level. So if you have high inoculum level, they die off slowly, as compared to they would die at a moderate or low levels in this case. Now, if you look at stainless steel, it doesn't matter. They die off at a very similar rate. So surface seems to matter in terms of survival of the microbes. But if you look at it, we are day 90 in your plant, so you've been running for 3 months, you still can find microbes on its surface. So once inoculated, these microbes can live in your surface for a much longer period, over a period of time. There may be some die-off, about 1 or 2 log, but depending on the initial inoculum that you brought in, there may be some survivals for an extended period of time. Now, the situation, this is in peptone, which is just a standard inoculum buffer in both stainless steel and plastic. But if you add the onion juice, it has been suggested that there are some antimicrobials in onion juice, that may do some inhibition there. To our surprise, we don't find that microbial inoculum being resisted. Actually, it became a lot more stable. Both the E. faecium and the Salmonella became a lot more stable in the presence of that onion juice that was deposited on the surface. So that residue is actually making it worse for it to die off. It's actually promoting it to survive for a much longer period of time. We have less than a log reduction in 90 days of Salmonella on a plastic surface in this case. So that puts a question into our mind to thinking about that if you have residues of juice or other things on the surface, those are the points that you want to consider. There are higher risks in those areas. if you happen to have a microbe land up there, it may survive better. So the next part we did was the transfer in dry environment. So before I talk about transfer in dry environment, how would a microbe transfer from a surface to a commodity of interest? And how would you control this process? Because you can throw something at a higher speed or a higher impact, and you may get a different transfer rate. So what we did was we actually designed a setup that allows us to control this experiment in a precise way. We have a platform, a mechanically controlled platform, where we can control the force and the time of contact and the frequency of contact. And we introduce an inoculated onion or a plastic surface on one side and a recipient surface on the other side. And we can mechanically control this device. And we can also do this with the peach. In this case, we have a whole peach here, the inoculated surface in this case. And we would do the experiment as outlined here, where the contaminated surface would come in contact with the onion skin, and we'll remove it and measure the transfer rates in this case. For the peach experiment, since the skin was not easy to remove, in the case of peach, we use a whole peach in this case, and would recover the microbes from the peach in this case. And we measure the transfer rate, which just tells us that how much population end up on the recipient surface compared to the donor surface that we start with. Okay? And here's the design of experiment. We chose a contact force of 10 newtons. That's about 1 kilogram of weight. 30 seconds of contact, 5 repeated contacts in this case. And we're looking at transfer direction, surface materials, strain, inoculation levels, as well as onion extract and soil water as an inoculum condition. How does that affect the transfer in this case? I just want to tell you that from the transfer direction, we found that onion to PU is higher transfer rate than from PU to onion. So PU is a plastic. So it transfers more efficiently from the produce to the surface than from surface to the produce. Okay? So once it lands on a produce, it has a less transfer to the other samples that are passing through it as compared to going from the surface, from the produce to the surface in this case. And we also found that the surface material did not matter in the transfer experiment. Okay? Whether you got a stainless steel or a PU, as long as there are no cracks and crevices in there, assuming similar quality of the product, the transfer rate was not affected by surface material. So I'm going to focus on the rest of the three things that are the strains, inoculum level, and the extracts. So if you look at the transfer rates, so this curve indicates the frequency of transfer, and on the x-axis we have a log transfer rate. What we find is that you have at high inoculation level, the transfer rate of E. faecium, which is the surrogate in this case, was higher than Salmonella. But at moderate to moderate level of inoculation, they were both similar. So Salmonella has a higher transfer rate relatively when it is at a moderate level as compared to when it's at a high level. So it also matters what level of inoculum you got on the surface. Now, interestingly, these transfer rates, I just want to point out that if you have water present, these transfer rates would go much, much higher. Okay, this is in a complete dry environment. So if you are even doing sanitation, you want to make sure that your equipment is dry when you run the product. So these transfer rates are about 1 to 0.1% transfer from the surface. But when you have onion extract or soil water present, your transfer rates go to about 61%. It's that sticky material that the onion extract leaves behind really makes it very easy to transfer back those microbes from the produce to the surface in this case. And that sticky material is something that you see on a PU contact surface like this. And in case of soil, actually, it somewhat impedes the transfer and has a much lower transfer than the other cases considered in this case. So was significantly impacted by the inoculum carriers. Now, I'm going to talk about a slightly different commodity, just to give you a sense of does it make a difference if I have a different kind of commodity running through my packing house? So now this is a slightly different process. As I mentioned, you go through hydrocooling, storage, washing, drying, waxing, sorting. Slightly different process, but there is a dry handling process starting from here once you start drying the produce, and there's almost a dry line here as shown here in this figure. We did the same other study as I described, and I'll move through in the interest of time here. So what we noticed was that at high inoculation level, it did not matter which strain we had on it. The transfer rates were similar between E. faecium and Salmonella. While at moderate level, we could not detect transfer in this case. There was no transfer to peach at moderate level of inoculation in this case of experiment. So we compared different surfaces. Again, surfaces did not matter between PU or PVC. If you got a different material, it did not make a difference in the transfer experiment. And interestingly here again, this is the uncoated surface, and if you have a juice left on it or a wax, because they do waxing in peaches, you get a much higher transfer rates than the control, which did not have that inoculum condition in there. Now, in this case, the juice was dried, so there was no wet moisture in this case. The wax is already dry in this case, but it tells you that the presence of these contaminating factors can significantly enhance the rate of transfer. And some of the images showing that what happens when you have a wax present and how that takes the fudge out, but also can enable the transfer of microbes. Now, if you compare peach, nectarine, and onion, I'm not saying they're all same, but in terms of transfer rates, there's not difference. So clearly tells you that microbe does not have any preference in terms of that transfer probability. If it is on the surface, it's going to transfer to your product. And what really makes the most difference is what is present along with it. So coming to the summary of the first part, first of all, we were surprised that it can survive for that long period of time on food contact surfaces under a very relatively low humidity conditions. The second thing is that if you have a produce extract like onion juice, it may help it extend the survival. May not increase the level, but maintain it for that long period of time. The transfer rate was not affected by surface material, but it was affected by bacterial strain, inoculation, and the presence of produce extract. I just want to emphasize that, and the presence of wax or extract will facilitate the spreading of pathogens in the facility by enabling better transfer between the surface and the produce. So in summary, developing effective cleaning and sanitation practices on food contact surfaces may reduce the risk of cross-contamination, particularly if you are targeting things like areas where you build up the organic residues, waxes, or any of those other things that you have with the juice and everything that would help reduce the risk. I'd also like to, do I have a couple of minutes? Yeah. Go ahead. Okay. I also want to talk about a slightly related area that we've been working on. So we've been thinking about how to actively manage this risk. So we can always do clean break and cleaning, and there's a lot that goes through that we clean, but there are a lot of facilities that we clean not that frequently. In some cases, we go for, as Laurel was mentioning, a full season before we have a clean break. So how do we manage those risks in some cases? So we've been working on this idea of, is it possible to develop coatings on legacy equipment? You can always buy a new equipment potentially which may have coating or some treatment done to it, but that's a lot of expense. So can we do something on a legacy equipment? And also, can we make these coatings with food-grade materials? Okay, so that it's easy to deploy potentially. Still will need to go through the approval and regulatory processes, but it's easier to deploy than something that is not food contact approved or food grade in this case. So this project was also supported by CPS and also previously supported by USDA before we got some funding from CPS to do this research. So the idea here was that can we select a biopolymer matrix, which are made up of chitosan, gelatin, beeswax, and zein, and find a way to deposit these matrix by easy methods, spraying, dipping, brushing, and something that where we can store common sanitizers like chlorine on the surface of it. So that we make these compositions such that we can actually bind chlorine to this surface and in the process stabilize the chlorine on this surface. So there are two formulations of it. One is a direct formulation where we introduce the biopolymers. In other case, we introduce what we call as chlorine-binding yeast particles, which are inactivated, and they bind chlorine in their core and form a coating. I'll tell you briefly about what we achieved with this approach. So in this case, for those of you who work with chlorine, it's always recommended that you make fresh batch. It's not storage stable. So here, after depositing coating, we could stabilize these coatings for a relatively long period of time, at least for 4 weeks, for a month in this case. And we did some dry abrasion and wet abrasion. These are the tests that were recommended based on the EPA guide for coatings. And we could see significant inactivation after a number of dry abrasion cycles. And based on the EPA guide, we think that these coatings can handle dry abrasion and some degree of wet abrasion for at least 3 weeks on consumer-grade surfaces. That's what the EPA test would recommend based on this. We also did some cross-contamination tests on diverse surfaces. Now, in food processing facility, not all surfaces are flat. We have surfaces that have something like foam. We got brushes. We got totes and bins with multiple crevices in them. So the summary of this slide is that in each case, on a coated surface, we were able to significantly reduce the risk of cross-contamination. So these were done in a lab environment. We said, OK, what would happen if we were to take this to a field study, like where we have in a processing packing house, and we were looking at a packing at that grading table, where you have the produce in this case that has gone through all the cleaning and sanitation processes, and that's the last step. But that was where it comes in contact with humans and all the other aspects to it, and there's no sanitation available. So we said, OK, so that was the interest that our industry partner had. We ran this for a 1 day of operation on a grading table. And the results were encouraging. We were able to reduce the total plate count on an compared to coated surface, reduce the coliform and the yeast and mold count. And these are all the microbes that are coming from the produce itself. There were no inoculation done in this case. We were looking at the natural microflora that would get transferred again. So things do get transferred because these surfaces were clean before we begin. So they were clearly sanitized. And on an uncoated surface, you can get about 4 log, close to that, and you have about 3 logs of coliform. And this product has gone through cleaning and sanitation before that. OK, so I just want to leave you with this thought. And so in summary, these are formulations which are rapid deposition, and they can be done on legacy equipment. They're made with food-grade materials. We have tested some residual chlorine on the product that they leave behind. There's not much that you can measure, as indicated here, of chlorate and perchlorate content. We did not see any perceptible color change in case of peaches because they come in contact with chlorine, so we were concerned that would that make a difference. And then the cost aspect of it, I just want to highlight how much was the cost. And this is the cost estimate based on us going to the market. This would be very different if a commercial vendor was to go in the market because they will buy in a bulk. This is my grad student calling up the companies and saying, OK, how much you'll sell zein for? And we are not a bulk buyer. So this cost estimate is just to give you an idea of what it would cost, but something that you can, if you're interested, could be considered. So with that, I'll end with thanks to the organizers for the opportunity to share these results, and happy to have your questions on any of the topics that presented.
questions for Dr.
Did I miss any specific coating names or brands or anything?
No, these are the ones that we made in the lab. So there's not a brand associated with it. No, they're not commercially available. No. Yeah.
So you're saying at this point it's about 4 weeks top that you're seeing that it lasts through, or possibly less than water? Is that correct?
About the antimicrobial coating?
Yes.
We tested for 4 weeks, and we did not see significant reduction in chlorine content. So it stabilized the chlorine content for that period of time. Now, it is possible the chlorine will get depleted with light. If you're in an environment where you have a lot of light coming on the surface, that will change. So again, our condition may not represent every condition possible out there. But if you prepare chlorine, you typically can't even stabilize for a day. So here we are going for significant period of time. So we think that this can work in areas where you cannot clean frequently, or even on surfaces may not even touch the product, but maybe in a zone 2 surface, where you have potential risk of some splash or other things. So these kind of coatings may work in those zones also.
But they were more, the 4-week period was for dry abrasion.
Yes.
Not the wet abrasion. The wet abrasion was the lower 3 weeks.
Was the lower 3 weeks, yes.
Did you also check this into different types of varieties in terms of short-day onions, long-day onions, and was there any effect that was noticed into these? It's general.
So we did not, we followed the yellow onions in this study for our transfer experiments. And we did not look into different varieties in this case. So one thing that is possible, because the yellow onion skin is slightly different than what you have on a red onion, that could influence the potential transfer in some cases. However, when we looked at comparing to peach and nectarine, which are very different, and the transfer rates were very similar across, so that would suggest also that it's more of how the microbes are present on the surface and what they're present with. That makes a more difference than the food contact itself. So it's an interesting question, but we have not explored that angle in terms of if there are finer differences between onion varieties in that sense.
What about with ATP testing with the different products? Does that affect the ATP swab numbers for food safety documentation?
In terms of the transfer experiments?
You have to swab for
Sure.
ATP, but if you're going a longer window without a cleaning process, would it also hold that back? Is that part of the results?
Yes, it would actually hold that back, and you'll have a higher count if you're accumulating more in this case.
Okay.
Yeah, because in our experiment, we're not accumulating. We are just inoculating one time and leaving it there. But if you are accumulating over a period of time, you would get higher counts. Because in your case, you're running the plant, as compared to in our case, we just inoculate once and just leave it follow it over a period of time. There's no other step where we're bringing any organic matter or other things in that setup. Yeah.
Any more questions?
No.
Thank you, Dr. E.
Thank you very much. Thank you.
All right, I got a couple of videos, so that's why I wanted to pull up my computer, and hopefully they'll run. Like Chris said, my name is Daniel Jackson, research manager at the UGA Ag and Environmental Services Labs. So my lab provides flavor and quality compound analysis for a variety of crops, but Vidalia onions are one of the ones that I work mainly on. And so today I'm going to present some preliminary results from a project we conducted last year and we're repeating again this year that I'm really excited about. And basically what we did is we characterized 10% of the entire Vidalia onion region last year, looking at soil profile and trying to relate that back to the flavor and yield of Vidalia onions. So just wanted to provide some kind of clips of this process. So I know most of the people in this room are quite familiar with the Vidalia onion industry, but But just as a reminder, I mean, these are short day Grand X type yellow onions that are restricted to a 20 area region in southeast Georgia with an annual farm gate value of $150 million. And Vidalia onions have built their brand around having a characteristically sweet flavor. And so protecting that brand and that flavor profile is clearly very important to the economics of this brand, this state, and this particular region within Georgia. And so when I think about flavor chemistry and what that means, really what we're talking about with onions are volatile sulfur compounds. And I know this is a big graph. It seems really complex. And so we're going to have a test at the end of this, so hopefully you'll be paying attention. But this diagram just kind of shows you the process and the complexity of the production of these flavor compounds. And so the green area here, that's an onion cell. And so within the onion cell, there's the vacuole, and within the vacuole, there's this allanase enzyme. However, when we cut open an onion or we bite into an onion, we break those cells and we rupture that vacuole. That allanase enzyme gets released. It interacts with these compounds, which we call the S-alkenyl cysteine sulfoxides, which then start this cascade of chemical reactions that produce these flavor compounds. And chief among these that we really care about is this one down here, which we call the lacrimatory factor. That's the compound that causes tearing when you cut open an onion. It also is the one that gives a lot of the burning flavor characteristics for onions. And so that's really important for pungency. Also thiosulfonates, specifically methyl thiosulfonates. These are the ones that we analyze that they give onions their metallic flavor, their grassy green, a lot of the aftertaste in onions. And then pyruvic acid is a byproduct of this chemical reaction. It doesn't have a flavor itself, but it's a really good indication of overall pungency because it's produced by this chemical reaction. And so by monitoring these compounds, we know the overall flavor and pungency of onions and can help monitor for the Vidalia onion crop. So when we talk about any sort of phenotypic response, so flavor is just another expression. Everything is a result of genetics and environment, right? So genetics we control by only growing specific varieties of Vidalia onions, but environment, there's so many other things that can influence flavor profile. And some of the things that have been published to be associated with flavor profile are things like irrigation or water content, root zone temperature, also nitrogen and sulfur fertility rates. So all of these things can influence the overall pungency of an onion. And then there's some work going back over 50 years. We've known that among these environmental factors, the most important one is sulfur availability and the amount of sulfur that we apply to our onions and uptake, because again, these are volatile sulfur compounds. And so when we think about sulfur, first off, it's a primary element. We need it. It's essential for any plant production. So it's something we have to have. If we have too little, we have deficiency symptoms, we stress, we have lower yields. However, particularly for onions, it's kind of, a little bit's good, but if we have too much, it'll actually excess accumulate, and that's when you get really pungent onions. You need a little bit, but you don't want too much, kind of a Goldilocks situation. And then, particularly here in our sandy soils, Randall et al. Back in 1995, working on Vidalia onions, found a linear relationship. So as you increase the amount of sulfur that you apply to onions, you see that linear increase in the overall pungency. So controlling sulfur is really important. And so, like I said, I've been working on onions for about a decade now. One of the projects we did a couple of years ago was looking at individual fields within the Vidalia onion region, knowing that there is some variability in soil characteristics, and seeing if those differences in soil characteristics within a given field were enough to influence sulfur availability or onion pungency and things like that. And so what we ended up finding is, yes, there did seem to be a trend. And what we found is kind of dictated by this graph, where onion yield, pungency, and sulfur content within the bulb seem to have a relationship with the clay pan depth. And so in our fields, a lot of times when we look in the profile, we'll have a really sandy surface horizon or topsoil layer, and then we'll have a pretty heavy clay pan at some point. And that can be anywhere from 6 to 8 inches all the way down to really, really deep. And so what we found is as you get deeper, you generally get sweeter onions. And the idea is clays really hold on to sulfur. And so if the clay is shallow, the theory is that the onion roots are able to access that sulfur deposit, become excess accumulate, and become pungent. And so as depth increases, pungency decreases, bulb sulfur decreases, and yields decrease. So other important variables that this study identified is soil texture. So sand and clay content are really, really important. Also organic matter, which allows to bind to sulfur, and soil pH. So it's all about basically the soil chemistry and ability to hold on to sulfur within the profile. And so that's really important. We know that variability within a field can change actual yield and pungency. And so we know that there's some variability, but the question is, okay, what's the variability across the region? Because that's really what we want to know. And so, the question we're asking, how much variability exists across the entire region, and how does that influence flavor at a regional scale? So here's an example. So here's 2 different soils that we commonly find within the Vidalia onion area. This is a Tifton soil. That's our state soil, sandy horizon up here, and then here's the clay pan about 8 inches down. This soil is a leaf-filled soil, sandy topsoil layer. This whole area right here is actually what we call an E horizon. It's all just sand. You don't actually start getting clay until about 35 inches down within the profile. And so these are going to hold nutrients, they're going to hold water very differently. And so what we saw previously is that influences flavor. But what we really want to know is if 95% of the industry is a Tifton soil, we don't need to worry about leaf-filled soil. We want to understand what is the variability and how to address consistency and pungency based on the soils we see across the industry. And so the first thing we did, I started looking at the map. The yellow area is the actual Vidalia onion region. And then I know you can't see all this, but this is just a map of the variability in soil types within a small section of the industry. So you can see that, we're not all just Tifton soils. There's a lot going on here. And so that's kind of the point of this project. And so the first thing we did is I went out and working with Chris, we interviewed 9 different onion growers. We selected 39 potential fields across the entire industry, and that represented about just shy of 2,000 acres. So that's 20% of the industry. We kind of mapped the fields. Based on that information, we then selected 13 fields across 6 different counties, and we selected these fields based on the varieties that were being grown and also the soil types. And so what we ultimately did is we decided to take one sample to represent 10 acres of area. And so this is a 140-acre field. You can see the soil types by the different colors. And what we did is we put 14 different points across this field to represent those different soil types within the field. And so ultimately, it's about 1,000 acres. About 97 soil samples or locations were collected across the industry. And so when I actually did the project, we created the map, had our points, we walked out to each one of the fields, we navigated to the points using our GPS, and of course my computer's running slow. Ultimately, what we did is we harvested the bulbs from the middle 2 rows within the field. So about a 4.5 foot section from the middle 2 rows of the field. Those bulbs we brought back to the lab for pungency analysis. We also did yield, and we did foliar nutrition. So there it goes. It looks like it's working. Then right in the middle of each one of those plots, we characterized the soil profile. So we used our soil auger, went down, and characterized the profile. And so here's what it would look like. So we recreate the profile. We measure the different depths. So in this example, we have 3 different soil profiles. We have an A, an E, and then a clay pan. And so we measure where those are. We took subsamples of each one of those soil types, brought them back to the lab for characterization of nutrients and other things. And so again, basically, we have yield, flavor compounds, and we're matching it to soil characteristics across the region. And so, results, what did we see? This is our first year doing this, and the biggest thing we saw is the diversity in soils. You know, I really found it striking. So here's our Tifton soil that we see a lot. There's another Tifton soil over here, but you can see they're quite different. I mean, that's a lot heavier clay, a lot more rock. It's going to hold nutrients completely differently. This soil, that auger is about 5.5 feet. I never hit clay in this soil. All the way down, just never hit clay. It's beach sand. That's going to hold nutrients very differently. This soil, it's gray down here. That's all clay. But it's gray because it's leached. It sits in water. So basically, this tells us that this soil has a seasonal high water table that's actually right in this area. And so a couple samples actually hit the water table between 10 and 16 inches down in the profile. Obviously, that's going to be very different than this soil is when we think about water and nutrient differences. So anyway, I could geek out on soils for a while, but I just wanted to show you some cool pictures. There is a lot of variability. And so, again, part of that interview process, we also looked at grower cultivation practices. And so I got some information up here. Sulfur applied and nitrogen applied. You can see that there's a lot of differences between what people are applying. Sulfur, one grower was applying 26 pounds per acre. Some others were up about 100 pounds an acre. So, you know, that's almost 4 times the amount of sulfur applications. Nitrogen, again, huge difference between nitrogen applications. Yield, clay pan depth, pyruvic acid, the crematory factor. So when we think about these two compounds over here, again, these are pungency. These are extremely sweet onions. These are quite pungent onions. The average is actually a pretty solid, that's a pretty good onion. So across the industry, this past year, we had a pretty mild and pretty sweet onions. So I thought that was pretty interesting. But we have some pungent onions happening too. So now let's try to figure out what's happening and see if we can find some correlations. And so from our variety trial work for the last 20 years that UGA have been doing this, we know that variety is a really, really important thing. So what I have here is I have pyruvic acid and the crematory factor by different onion varieties. So these are all the varieties that I tested across the different fields. The points represent the data. And what you can see is there's definitely some differences between some of the varieties. Obviously there's some overlap, but there's definitely some differences between varieties for their overall pungency. However, we know sulfur availability is really important. We found no correlation between sulfur content, again, p-value of 0.36 and 0.38. So sulfur application rate wasn't driving pungency in this case. Yeah, Cliff? This is application rate. So this is sulfur applied fertilizer pounds per acre. So this is what the growers told me they applied. Okay, so application rate of sulfur. And then, but you can see differences in variety in here too. So it's a complex system. This is pyruvic acid, the crematory factor by soil type. So soil series down here. What you see is there's a lot of variability in here. Seems to be there might be a little bit of difference, but overall, they're all kind of like right here in the middle. You can see, like, for instance, down here on the Tifton, again, that's our state soil, you can get really sweet onions, you get really pungent onions. So we can say that's not necessarily the driving factor. This pyruvic acid and the crematory factor, looking at clay pan depth. Remember my previous slide, or the kind of hypothesis, we found this association where as you get deeper, we should see a trend. So in general, there is a trend going this way, but it's not statistically significant. And maybe that's just this season or something like that, but it's not a statistically significant trend in this situation. So there's more going on. It does look like it could be a factor, but it's not an overwhelming trend. Interestingly, this is something we haven't seen a whole lot of, is pyruvic acid by nitrogen fertilization rate. So this is the amount the growers reported they applied, 75 up to, I think it was 160 pounds. We saw a very strong, statistically significant relationship between pungency and nitrogen application rate, but no difference in yield. I just wanted to point that out. That's completely flat. So more nitrogen does not necessarily mean more yield in this case. Another thing I thought was pretty interesting, poultry litter. We had a couple growers that were saying they applied poultry litter, statistically significant. If you increase poultry litter application, or basically most growers weren't applying, some growers were. If you applied, you tended to have higher pyruvic acid, lower yields. Again, I'm not claiming that that's causing. I just wanted to show that there's a lot going on in these systems, and that might be something to consider. So as you noticed, we've got a lot of variables that are competing to flavor. They're all interacting. And so even though this is preliminary data, I wanted to kind of look at this from a more machine learning, larger statistical approach, because I can't comprehend all of those different variables and how they're competing. And so I wanted to just do a machine learning algorithm to try to predict what are the main variables driving onion pungency in this system this year. What it identified is nitrogen application rate, foliar sulfur, sulfur within the A horizon or topsoil. These are the main things that are actually kind of predicting onion pungency. And then when I take it one step further, it created this, what we call a conditional inference tree. What it's doing is it's basically segregating the data based on pungency and saying that fields 1, 3, 9, and 12 have some sort of characteristic that I'm not necessarily measuring that determine pungency. Then all the other fields, the most important factor was nitrogen application rate. And so you can clearly see there's kind of, it's just basically, it's a machine, it's just a computer algorithm trying to figure out how to partition the data. I just think it's really interesting. Next year, we're going to repeat this study. We'll get more data and try to see if we can use this model or refine this model to get a better prediction. So anyway, again, preliminary conclusions. Number one, there's a substantial amount of variability that does exist across the Vidalia onion region. That's really important. Onion pungency appears to be a relationship between variety selection, which we already knew, field characteristics such as soil, sulfur, clay pan depth, organic matter and texture, also nitrogen and sulfur fertilization rates. And then again, we're going to repeat this work in this coming year, hopefully add more to this and see a seasonal response. Obviously, all of this work is funded by the Vidalia Onion Committee, so I really appreciate their continued support of my lab and this research. Also, without the participating growers in this program, we wouldn't be able to do this. So thank you for the committee, thank you for the growers, and I also want to thank the field crew, Guy Euro and Matthew Thibodeau. This was a lot of work. So we were hauling, hundreds of pounds of onions across fields. So I really want to thank them for all of the work. It was a lot of work, but it was a lot of fun too. And then finally, I've created a project website for this. So if you scan this QR code, you can go check out our website and kind of see updates on what you're doing or what we're doing, and some more kind of just photos of what we're about. Also made a little YouTube video to describe my methods and stuff. So with that, I'll take any questions. Yep.
How much variability in the soil organic matter was it, and was that a direct reflection off of the litter that was applied, or was that based on the crop, previous crop?
It's a great point. It's not a direct result of litter application in this season, but it can be highly variable. I don't remember the numbers for this project, but I remember the project I did a couple of years ago. We had we had organic matter anywhere from like 0.5% to almost 3% in different onions fields. And we always talk about we're in the southeast, we don't have a whole lot of organic matter. Some of these soils are holding a decent amount of relative, like 1% up to 2% in that clay pan, and especially if we're rotating with other crops and applying chicken litter. I think you.
So would the organic matter correlation, would it be positive or negative in terms of flow?
Probably positive, but I speculate that it's positive. I don't know the full data, but it's saying that that's an important factor. Organic matter is one of the key things that holds nutrients. If you and holds sulfur particularly. If you remember my leaching study a few years ago, basically organic matter was one of the few things that allowed the soil to retain sulfur. If you had low organic matter, it's going to leach. It's going to leach sulfur within a relatively short time. If you have organic matter, it's going to hold it for a long time. So, yes, sir.
You have the one category that you had, I guess, located the variable, like working.
Yeah.
Have you looked at, you said some of the soil samples were rocky. Have you looked at like the mineral content?
So I haven't looked at rock percentage or anything like that. That is the next step. You know, like Tifton soil. You know, that's just a name, and that's what I use to plug in my model. But if I go to the NRCS website, I can get a lot more information than just Tifton soil. You can get rock percentages, you can get, you know, saturated conductivity. You can get all sorts of information about what a Tifton soil is that is a lot better for putting into an algorithm to actually help you predict. So that's a lot to say that we can use more than just a name, and I plan to do so, but I thought this was a good way to just kind of 1 year worth of data, see that we're going beyond just one variable, here's a relationship type thing. So.
Is that from the soil survey?
Yeah, all this data came from the USDA web soil survey downloaded, so. Any other questions? Yes, sir.
So, you mentioned that the pungency is highly dependent on genotypes and environmental interaction.
Yep.
So based on that, your observation, that how much is genotypic impact is super important for these, or the environmental impact is super important for these?
Yeah, so there's some research going back from, I think, the 90s that said genetics are 80% of pungency, environment's 20%. But we're already controlling the 80% because we have an approved list for Vidalia onions. Only specific varieties can be grown as a Vidalia onion. So we're kind of already taking the genetic component out, and then we're just focusing on that last 20% because, again, we want consistently sweet onions.
And
So,
one more
sure.
So on this current place, do you see that there is a need that we need to do something to help the growers?
So you're asking about do we need sweeter varieties or do we need different varieties, I think is the crux of it. I'll just say that there are differences between varieties and their pungency. Some are fairly consistently sweet. Some have the opportunity or the potential to become more pungent. And so there are definitely, from a pungent standpoint, there are better, more consistent varieties as far as sweetness. We do a screening every year and we publish it in our variety trial handbook, or our research update that Chris puts out every year. So we have that information going back like 15 years. So, yeah. Okay.
Any more questions?
Thank you, Daniel. Okay. As I mentioned before, I'm Chris Tyson. I'm the area agent for onions in the Vidalia region, and I'm also the superintendent at the Vidalia Onion Research Farm. So before we dismiss today, I just wanted to give sort of a brief crop update and hit a few points on disease management that I thought might be important today. So anyway, I wanted to start off by mentioning our onion blog. There's the website right there. And you can visit this blog and sign up to receive updates. Anytime we have a meeting or some kind of news pertaining to onions, we'll post on this. So if you don't already receive updates from us and you're interested, just go to the website there. You can search, also just search for UGA extension onion blog, and it'll usually come up. So also want to mention coming up at the Vidalia Onion Research Center on January 23rd, we're going to have a more in-depth disease management update with Dr. Bhavesh Dutta, and we're going to have a low country boil that day. It's going to be good, so come on out if you can. But the 23rd, that's a Tuesday. We're going to start at noon. So we're going to give another sort of crop update. We're going to have in-depth disease management update from Dr. Dutta, and then if weather permits, we're going to have the opportunity to walk through the variety trial and see what it looks like at the early stages. Most of the time y'all see it at the field day when it's, a lot bigger, but if you want to get an early look at it, we'll try to do that. We've got 53 varieties planted this year, so come join us on the 23rd. So I've been out with the extension agents, the county agents, to look at the crop a little bit the last couple of weeks since we came back from Christmas break. I believe this field, this is somewhere around Glenville in Tattnall County. This was on January 3rd, actually. And there's not to most people, there might not be a lot remarkable about this picture, but to me as a agent and to some of you as growers, if you have any memory of last year on your mind, last year we had a hard freeze in December of 22. We lost a lot of stand in our crop. The most significant thing I'm seeing this year is much better plant stands, really great plant stands. Most of the fields I've been in have really good stands. So I'm glad for that. I know the growers are too. And two, I really feel like a lot of growers this past planting season have really, since we had such trouble with the cold killing our stands last year, a lot of growers really focused on doing a better job planting, slowing down planting, transplanting, and really focused on the quality of their transplants. And I definitely think it's paid off this year. So a great stand here. I don't know what variety this is, but here's some onions that were a little bit bigger. I think this was taken on January 3rd also. So we looked at some younger onions that were planted right before Christmas too. Most of the time when I take a picture of an onion field, you can't really tell whose field it's in, but some of you might recognize this bed pattern here, one of our growers here. So he was in here earlier, but I think he left. So, but this field looks really good too. A great stand. Here's a few of some bigger onions we looked at just this week. Some Japanese onions. That's about some of the biggest ones I've seen around. So, but overall, I feel really good about the crop at this point. We are sort of off to a cold and wet start, and we may be in a pattern. We're in an El Nino year, I believe, and that can make it cooler and wetter. So that trend could continue. We don't really know, but it could. I think we're already kind of to the point where we may start seeing some delays in getting fertilizer and fungicide out because it's too wet to get in fields, and we're waiting on fields to dry out. So hope that's not a continuing trend, but it's a possibility. So transplanting went pretty good this year. Our foliage growth to start off is fairly slow, just basically because it's just been cool weather since really about the middle of November. It's just been pretty cool or cold, you know, and the crop hasn't put on a lot of foliage growth, but that's okay. I don't think anybody's really complaining about that. We have a lot better stands though. So if you look at the heat, the growing degree days, the heat unit accumulation, this bottom line right here, that's us this year. And this is measuring with a base temperature of 50 degrees, starting from November 20th up to, well, it goes up to the 10th when I pulled this graph, and then it forecasts out to about a month out. But this bottom line is us. So you can see it's, this next line in the middle here is the average. So we're a little bit below average. This top line right here was last year. You see last year where it's just flat right here, that was that really cold period around Christmas time when we had the really hard freeze that killed a lot of the stands. We didn't accumulate any heat during that period, but and then after that we had a good bit of warming weather. So we're a little bit below average, but you know, I think we're all right. So I wanted to talk a minute just briefly about the Bravo situation. I know the Onion Committee and I think Cliff or Chelsea sent out some information about Bravo. EPA's put out what they call a PID, or a proposed interim decision, to review chlorothalonil, Bravo. And basically what this is doing is they've proposed reductions in the amount we can apply to the crop. And if there's a lot of crops we use Bravo for, a lot of them it cuts the use by about half. So I kind of wanted to take a closer look on what this meant for us, for onions. This chart right here shows this is the proposed, the proposed rates. And if you come here, you see there's a lot of crops here, but we got two sections for onions dry bulb, and then onion like green bunching. I'm looking at the dry bulb because that's really what our production is called, dry bulb production. They're wanting to limit it to 9 pounds of active ingredients per acre per year. So we're currently, this is actually, this slide right here is a Bravo weather stick label. And if you look at the limits current limits for Bravo, it's on the Bravo label. Their limit is 15. So we're currently at 15, and they're proposing to cut it to 9. It's not quite half, but it's a pretty big reduction in what we can use. So you see here with the current Bravo label, we're limited to 15 pounds. That's where it currently stands. And that's basically equals 20 pints of Bravo used in crop during that season. So let's do a little bit of math here. If this proposal passes, it would reduce it from 15 to 9. So basically from 20 pints to 12 pints per acre in onions. So there's about 3/4 of a pound of active ingredient of actual chlorothalonil in 1 pint of your typical Bravo formulation, like Bravo weather stick. So we can use, the rate that we can use is 1 to 3 pints per acre. A lot of people, I don't hear a lot of growers using that 3-pint rate. I feel like a lot of people are somewhere between a pint and a quart, 1 to 2 pints. So, but anyway, if this passes, that would limit you to 4 applications in crop at that 3-pint rate. It would limit you to 6 applications at a 2-pint rate and 12 applications at a 1-pint rate. So that's kind of the rate reduction that you're looking at if this goes through. So I believe that there was a comment period open, and they said originally it was gonna, like December 18th was the cutoff, but I believe they have if I'm reading all these legal documents right, I believe it's been extended to January 17th. And this is actually the website here that you can go and make a comment as a grower and say whether you don't like this or you like this, or what Bravo means to your crop or chlorothalonil means to your crop. So I was just looking at this website, I think just yesterday. This is what it looks like. And if you click on the website, it's got a box right here for comment. And you can click on that and you can put a comment in there. And you can also see, you can click right here, posted comments, and you can see some of the public comments. And when I looked at it yesterday, there was only 29 on there. But what I was glad to see, though, of those 29 comments, I noticed that there were 2 or 3 Vidalia onion growers that had gone in there and made a comment about chlorothalonil in this proposal. So if it matters to you, if it's important to you, I would suggest you check this out. That's the website right there. So, Babesh, do you have any updates on this?
No, I think the Georgia Vision Commission is going to draft their own. So they're drafting one. I don't know when they're going to submit it, but they will.
Okay.
Submit it.
Okay.
Is committee going to submit one? Like, Vidalia committee? So, and there's also the business council. And also, there's a consortium of extension specialists in the southeast. We are doing one draft and we're signing it on the specialist assignment.
Okay. Okay.
So that
Good.
will go through IR4 southeast.
Okay.
And
Yeah.
I'm going to do my own also.
Yeah. And if you're a grower, or anybody can make a public comment. So, yeah, I was glad to see there were some Vidalia onion growers that had posted individual comments on there. So. We're going to talk a lot more about this on January 23rd at our update, but I wanted to give you the most recent ratings or rankings for disease management. Here's our most recent botrytis rankings here by these different fungicides. And these are from Babesh's slides from his data. He's got Fontellus Maravon marked in red here. And you said that was for Stemphil. It's
Stemphilium, it stopped working.
Mhm.
And Maravon has shown a low level of efficacy last year.
Okay.
So there's already a resistance in New York and Pennsylvania with Maravon.
Mhm.
But I think it's coming here in Georgia.
But you still feel like it's holding its own for botrytis control.
It's still there, but
Okay.
our growers use Fontellus early on salt spread
Mm-hmm.
for retailers that has more say over there, Fontellus. But Maravon is going down in 2-3 years. I would say it's going to be the same as well as Scala is.
I don't see a lot of Maravon used, but I see a lot of Fontellus being
Right.
used. So, okay. This is for botrytis. This is the rankings for botrytis if you need to take a picture or take notes. Stemphilium. Here's where everything ranks. You can see some of this stuff here at the bottom that we don't have efficacy on or it's slipping. Looks like the top rated ones would be Miravis Prime, Inspire, or Luna. And then followed by Quadristop, Pristine, Switch, and Quadris. So. Downy mildew. Let's talk about downy mildew for a minute. We've typically relied on Omega, Arondis, some maybe Zampro, and then we use a lot of Bravo and phosphates. good economical products to use for downy, help with downy. We were able to get in a field study last year in a grower's field where we had some downy last year. County agents in Tattnall County conducted that trial, and previously we hadn't had Ridamil or Ridamil Gold Copper in our lineup. Babesh has got it added in this year. It's in red, and we've just got kind of 1 year of local data with this, but I think, wouldn't you say it would be a good something to maybe to use this year or consider using?
Candidate, you know, with downy, we don't want downy, but for pathology, you have to assess all these fungicides we need the infection. So this is the first time Ridamil Gold Copper worked, and Jason was part of the study, and he had also watched for it. Did well, out of our expectation.
Yeah. So it did well in our local field trial this past year. And I believe it's used a lot in other regions for downy control. And for whatever reason, it hadn't done very well for us in the past, but this past year it did good. So
what I was saying with that, the first year we had downy, which was Langston was 12. Yeah, 12. Langston was still here. We tried Ridamil Gold Copper, but I think in the grand scheme of things, that's when we first saw that reason seemed to help and work somewhat. We tried Ridamil Gold Copper because, again, that's what they sprayed a lot in the West. And we didn't have it work, but again, part of the problem also was we call all this late. And if you let downy really get ahead of you, it doesn't matter what you spray. The weather doesn't help you and you don't get out there timely, it's going to get you. This is all there is to it. You can't get behind the 8 ball.
Might end up with Ridamil Gold problem.
Yeah.
And it worked in Tattnall at that trial, but not in two other trials that didn't work. Xanthro was still the experimental number, the reason was the only one at that time.
Seemed to do anything.
And we had in the recent years, we'd sort of taken off reason from our recommended list, but the feedback from growers is that y'all still believe it's got efficacy and it still works. We didn't have it in that trial last year. I wish we did, but going forward, if we're able to get it in a field trial, we're going to evaluate it again and see if it's something that we can use. So anyway, I know there were still a lot of people spraying it reason last year. As far as bacterial disease management goes, all your copper sprays, for the most part, Babesh has evaluated most every product you can think of. They're all very similar, not really any statistical differences, whether you like one over the other. You've got copper, and you've also got LifeGuard now. And most of you are using LifeGuard. There's been, I get questions, can I tank mix it? Do I need to spray it by itself? They've done some work with that. I will say it's probably preferred to separate them out, and you can actually stretch out your program a little bit longer, but if you can tank mix it too. You can tank mix the LifeGuard and the copper. There was always a question about, copper kills bacteria, and LifeGuard is a live bacteria that you're spraying. Would it have an antagonistic effect on the LifeGuard? If you talk to the company folks, they say that you can tank mix it, and preliminarily from what we've seen, you can do that too. Correct, Babesh?
Yes.
Okay. So
it does kill a little bit of LifeGuard, but not enough to cause it.
So either way you want to do it is really, I don't have a preference. Whatever you feel comfortable doing, you can do it either way. You can mix them, you can separate them. So, just wanted to mention real quick, we've been looking at neck length clipping at different neck lengths for a reduction in center rot or internal rot. This is a big issue with us at harvest, post-harvest. We got another year of data with this past season. and it showed that basically the longer you can clip that neck, the better off you are. And when I say longer, we just don't want you to clip it at the shoulder. And preliminarily, it looks like somewhere around 2 inches is a sweet spot, to reduce center rot. I've got a poster outside, right outside the wall here, or right outside the door here on the wall here, showing a little bit more of this data and also some other cultural practices along with the neck length, how short you clip them, looking at machine harvest and digging methods, and check it out right out here before you leave. But I just wanted to mention that we got another year of this neck length study data, and it's backing up what we've been saying. So and I've talked with a lot of y'all, a lot of growers, and I feel like there's several of y'all that have made attempts or made strides to train your crews to maybe cut the necks a little bit longer in your onions, and I think that's a good thing. So but anyway, that's really all I wanted to share with you today. I would like you to come join us on the 23rd if you're around. So at this time, I think it's about lunchtime, I will entertain any questions that anybody has. Anything I didn't cover?
I have one comment.
Okay.
Regarding neck length, this particular study and this particular outcome has been adopted by Washington growers, Pennsylvania growers, New York growers, and also Texas growers. You may see longer yellows, whites, green, reds.
I know you had a picture at the meeting back in September. You went to Publix in Tipton.
Right.
You found a bag of Vidalia onions, and I think it was in the summer. And the necks were poking out of the bag. They were cut longer. You can see.
2 inches of them.
You've also found some red onions, like that too. So I think that's a good sign to see that, and we think it helps. So
They also think it helps. That's why they started doing it.
yeah.
There's an outcome from body image.
Yeah. Well, I have to give credit for this. I've helped with this neck length study, but we did a preliminary trial with it a couple of years ago and saw some interesting results. And Ross Green in Handler and Evans County has sort of took the lead looking at this a little bit closer the last 2 years, and it's been a good data, good report. So.
Can you just make one comment on our new this mechanical harvesting grant which you got?
Yes. Well, what you got? Babesh is awarded a USDA grant to look at mechanical harvesting. It's over like the next a 4-year project. We're going to do some stuff the first couple of years, like looking at varieties, and because there's not a lot of machine harvesting by that area. We got one grower that's doing it probably exclusively. We have some other growers who have machine harvesters, but they're not necessarily using them. And if they are using them, they're not using them a lot. There's a lot of things we worked out. I mean, a Vidalia onion is a different onion than a storage onion out west that they use these harvesters on. So to start off with, we're going to look at varieties, and we're going to look at bruising and impact, and a lot of different things. We're going to sample all the varieties we grow this year and evaluate them for, impact. We're hopeful that we can get in the field and get maybe some growers to help us that have a harvester crank it up for us if they haven't used it in a while, maybe use it in a spot and let us run we have like a simulated onion, it's basically like an onion bowl, but it's got a lot of sensors in it. We want to run it through the harvester and measure the impact that it goes through, because as we all know, Vidalia onions are a little bit more sensitive to bruising than a storage onion that's grown somewhere else. So it's really exciting to see us getting this grant, and I think it'll be a good thing to help us look a little bit closer at it. Hopefully we'll get some good information out of it. Is there anything else you want to say about it?
No. You will be able to see robotic Vidalia onion. No. We'll pass it through those machines and see where the weak points, and then we'll evaluate the varieties that we'll look at. That will actually push on those harvesters where are the weak points. Okay, where are you getting bruised? And then we're gonna look at the varieties which are suitable for those harvesters.
Yeah.
So Vidalia varieties. So I think that's a great.
We've got a grower that's doing it now, and I think you have to modify your growing practices, your cultural practices a little bit, but it can be done. We want to see if we can help other people that might want to do it, help them out. So, all right. Well, anything else from anybody? Well, thank y'all for coming today, and hope to see y'all on the 23rd, and let's go get some lunch.